Endocrine cells receive information about the external and internal environments. We'll examine some examples where the need for additional blood glucose is regulated by different hormones and by different mechanisms. Sometimes an endocrine cell is also the sensing cell. In other cases, the information is relayed to the endocrine cell in a variety of ways. In the body, nerve signals directly stimulate the secretion of the hormone epinephrine in the fight or flight response. For example, a deer's sensory systems send a nerve signal to the brain announcing danger. The brain triggers sympathetic neurons to stimulate the adrenal medulla, which releases epinephrine. Epinephrine binds to receptors on the surface of many cell types to prepare the body for quick action. By way of second messengers, epinephrine activates enzymes in the liver to break down glycogen and fat cells to release fatty acids for energy. The heart beats faster and stronger, and the smooth muscle of blood vessels that supply the intestines contract. Blood glucose levels rise, and the delivery of blood glucose to the muscles and brain is maximized. Some nerve cells secrete hormones directly. When stimulated, certain neurons release hormones called neurohormones. Many of these neurons are found in the hypothalamus, and their axons usually terminate in the pituitary region. Here's an example. During finals week, you may experience prolonged stress and anxiety. This longer-term stress is conveyed to neurons in the hypothalamus of your brain. These neurons release corticotropin-releasing hormone, or CRH, into the bloodstream, which serves the anterior pituitary. There it binds cells that have its specific receptor, causing the release of adrenocorticotropic hormone, or ACTH. ACTH is a tropic hormone that causes the synthesis and release of another hormone, cortisol. ACTH travels via the blood throughout the body to the adrenal cortex, where it binds to cell surface receptors and initiates the synthesis and release of the steroid hormone cortisol. Because cortisol is lipophilic, it can enter every cell as it travels in the bloodstream. In target cells, like liver cells, cytoplasmic glucocorticoid receptors are bound to proteins. When cortisol binds the receptor, these proteins release, allowing the hormone receptor complex to move into the target cell's nucleus. In the nucleus, like most lipophilic hormones, cortisol receptors will bind as dimers to DNA. The hormone receptor complexes bind to specific regions of genes called response elements. Within minutes, the transcription machinery is assembled and gene products are made. Cortisol maintains proper levels of energy substrates partly by increasing the production of enzymes that synthesize glucose. This helps provide the body with adequate levels of blood glucose during times of prolonged stress. Our body needs an adequate supply of blood glucose for maintenance and occasional bursts of activity. However, too much blood glucose can be detrimental. Although the way our cells sense blood glucose levels is not completely understood, a low-affinity glucose transporter might be a glucose sensor in pancreatic beta cells. Glucose sensors in the hypothalamus, brainstem, and liver also play important roles. These sensors guide the interplay between two antagonistic hormones, insulin and glucagon, to maintain proper blood glucose levels. Insulin is secreted by pancreatic beta cells located in cell clusters called islets of Langerhans. Insulin receptors are found on almost every cell. Proteins in target cells are modified to prevent the synthesis of glucose and promote the synthesis of glycogen. Insulin also stimulates enzymes in fat cells to synthesize fats from fatty acids and glycerol, and stimulates protein synthesis in liver and muscle cells. The action of insulin is terminated by endocytosis of the hormone receptor complexes, and the number of receptors can be downregulated upon continued insulin stimulation. Glucagon is secreted in response to low blood glucose levels. Made by the alpha cells of the pancreatic islets, glucagon acts on the same targets like the liver, but with the opposite effects of insulin. Glucagon binds to a G-protein-coupled receptor. The resulting cascade of second messengers leads to modifications of proteins to break down stored glycogen to release glucose. It also stimulates glucose synthesis in the liver and kidneys. The release of glucagon is inhibited by insulin, which rises when glucose levels in the blood are again high. Insulin, glucagon, epinephrine, and cortisol can all affect the level of glucose in the blood by different mechanisms and in response to different stimuli. The absence of an insulin response results in the disease diabetes. Diabetes can be caused by a defect in the production of insulin, or defects in the receptor-mediated response to insulin. These graphs represent insulin, glucagon, and glucose levels in the blood of two people during a short fast. Click on each graph to see what happens when they drink a sugary soda. Can you tell which person is diabetic? Yes, good job. Sorry. The diabetic's blood glucose level remains high for much longer than normal. If left untreated, gradual damage to organs such as the kidneys and eyes will result. We've seen how different hormones affect the amount of glucose in the blood. Hormones can work together to coordinate the response to the many demands made on our body.